you very much for the introduction, and many thanks to the organizers for inviting me to speak at this wonderful conference. Hopefully you can see in my screen now. As I say, it's a great honor for me to speak at a conference dedicated to the memory of Nikolai Timofeyev Rysovsky. Most unfortunately, I have never had a chance to meet him, although I have missed my chances. I'm old enough to have had this opportunity. But the important point here that just occurred to me is that in the 1930s, a little less than 90 years ago, Timofey Rysovsky and his colleagues, Zimmer and Delbrück, were more or less the first scientists to obtain, through very ingenious experiments, reasonable sizes, reasonable estimates for the typical size of a gene. Now, we have gone a long way towards understanding genomes, genetics, and I dare say some steps towards understanding the whole universe of genomes, far cry from estimate of the size of a single gene, but these were the essential beginnings. So I think Timofey Rysovsky would have been amazed to see today's developments. I titled this lecture rather immodestly, Structure and Evolution of the Genomic Universe. Of course, I cannot truly give justice to the subject, both because of the lack of time, but more importantly, because of the lack of true understanding of the genome universe. So I will show you just some glimpses of the organization of the genome space, genome space time, if you will, that come from our work over years. So the beginnings of comparative genomics have been very humble in a sense. About 25 years ago, a little earlier, in the laboratory of J. Craig Venter, these two small bacterial genomes have been sequenced, Haemophilus influenza and Mycoplasma genitalium, both mildly pathogenic bacteria in human, possessing some of the smaller genomes among cellular life forms. So that feat, because at that time it was a very, very remarkable achievement. It looks humble 25 years later. So inspired by this feat, my colleagues and I immediately undertook a comparative analysis. They were very lofty goal to derive a minimal genome for life. So we derive the composition, the set of genes that would be necessary and sufficient to maintain the simplest cell. How do you do this? It's actually conceptually very simple, although it was not particularly easy with the computational tools available at the time. Basically, you compare the genes of these two bacteria, the small, simple bacteria with small genomes, and establish what is called orthologs relationships. That is direct correspondence between genes that have evolved from a single common ancestor in the last common ancestor of the compared organism. Simply put, the same gene in two different organisms. But then, of course, if you examine the list of orthologs between these two bacteria, which consists of about 200 genes, you see some gaps among the essential functions. Because of the phenomenon with entitled non-orthologous gene displacement, the situation when an essential gene is displaced by a distantly related or completely unrelated gene performing the same function. So examining less formally the remaining genes, we make some additions to the list of orthologs accounting for non-orthologous gene displacement, and then also informally eliminate some genes that seem to be specific for parasitic lifestyle of these bacteria. It might not be essential for a minimal cell. So this resulted in a theoretically derived set of about 250 genes that we thought might approximate the genome of a minimal cell. I think a variety of small additions, corrections, these estimates still stand and became the basis for a whole little scientific industry, so to speak, experimental studies of putative minimal cells. So this is the first very humble foray into the genomic universe that I wanted to introduce. Today, however, is very different from the 90s. And since that very time, since 1995, then these first genomes have been, of cellular life forms have been sequenced, the growth of the genomic library or the collection of sequenced genomes has been exponential. So if we look at this curve, the green one here, showing the accumulation of complete assembled genome sequences in the beginning of this year, there have been about 10,000 of these, but actually the real genomic library of prokaryotes, by the way, I maybe failed to say this, today I'm discussing only the relatively simple and small genomes of bacteria and archaea, collectively known as prokaryotes. So this is not the entire collection, because predominantly these days, and for the last few years, the sequencing effort has been directed not into obtaining complete genomes, but to obtaining genomes somewhat small or lower quality, either large scaffolds or shorter contigs, but of much greater quantity and diversity filled by the advent of next generation sequencing. So we now have already up to 1 million of diverse prokaryotic genomes to analyze, allowing a variety of conclusions to be made. So, however, I will now return briefly to the 1990s and introduce the approach that we have developed with my colleagues and that has become important and popular in the entire comparative genomic enterprise. This is the derivation and analysis of the so-called clusters of orthologous genes, COGS for short, that represents set of orthologs, as I introduced it, genes derived from the same gene in the last common ancestor of the compared organisms, systematically organized into groups, also taking account of gene duplication. These cogs have become one of the main parts of the comparative evolutionary genomic framework. What is important to note is that the cogs are evolutionarily stable by and large. They reflect, of course, very different evolutionary depths, but in as much as you can identify objectively a set of orthologs, they represent orthologous lineages, distinct units of evolution. And what we see, this is a figure from about 10 years ago, and today these blue areas would have been somewhat bigger, but not by much, actually. So what we see here is that the majority of the genes in each sequenced bacterial or archaeal genome can be included into such cogs. In other words, genes are highly conserved, resilient in the course of evolution, and you, for the substantial majority of them, you can trace the evolutionary history. This is entirely non-trivial, was not quite anticipated until genome sequencing started in earnest, and represents a cornerstone of the evolutionary genome. Because of this resilience of genes, we can understand something about the organization of gene universe and its evolution. So the dynamic genomic universe, a phrase that I want to use, is genomes in turmoil, because as we shall see, the genomes, at least of prokaryotes, but not only, are highly dynamic. So nowadays, we actually rarely speak of individual genomes. In a way, this can be considered a paradigm shift in genomics, and I dare say evolutionary biology. In the late 1990s, when molecular phylogeny started, with the seminal work of Carl Woese, the evolution of life has been studied by constructing and analyzing phylogenetic genes for individual genes. For instance, and most importantly, universal genes such as ribosomal RNA. In the 1990s and the early 21st century, the way to understand the evolution of genomes was indeed comparing individual genomes and tracing the changes that occur in them against the scaffold of such phylogenetic trees. Now, these days, things have changed methodologically, and our thinking has evolved. Thanks methodologically, again, to the rapid advances of next generation sequencing. And nowadays, we talk about, in evolutionary biology primarily, not about genomes of individual microbial cell, let's say, something you can isolate from by growing a colony of bacteria and archaea and sequencing the genome, but rather pangenomes, the entirety of genes that can be found in all isolates of a particular species of bacteria and archaea, setting aside the difficulties in defining the species, let us say in Escherichia coli, all the genes that you can identify in different strains and isolates. We shall talk in a moment about the typical structure of pangenomes. Right now, I want, however, to bring your attention to, so to speak, the dark matter of genomes. Indeed, in all bacterial and in nearly all bacterial and archaeal genomes, there is a substantial fraction of what may be called orphans. The substantial fractions 10 to 15, sometimes up to 20% of what may be called orphans. This is, of course, a pun, a wordplay on open reading frame and orphan, and these are genes for which homologs cannot be identified, except possibly in very closely related isolates. Apparently, and as we shall see, very fast evolving genes that may represent a reservoir of genetic novelty. So, having in mind the option of the notion of orphans, let us consider the organization of pangenomes, which is rather remarkable, because when genomes of a particular species, so sometimes a larger group of bacteria and archaea, are sequenced starting 1, 2, 3, going to hundreds, as done for well-characterized organisms, a remarkable picture emerges. Very quickly, with sequencing just a few genomes, you can identify the set of core genes, the core genome, relatively small number of genes, somewhat below the typical genome size, like 2,000 genes for most, give or take, for most bacteria and archaea. What happens then when you sequence more isolates? For the majority of bacteria and archaea, you keep seeing more or less linear accumulation of genes. That is, new genes are added. About that type of fraction, 10 to 15%, are added with each sequenced isolates. These are so-called open pangenomes because we do not really see signs of saturation for many of them. Some bacteria and archaea, notably, however, are different. They have closed genomes, such that you don't really see many new genes arriving with the sequences of new isolates. That's shown here for Bacillus anthracis, a major human pathogen. More recent data, we can still see that the majority of microbial genomes, microbial pangenomes are open, like here, and they more or less, the accumulation of genes more or less follows the power law, but with different exponents. So they grow at different rates with sequencing of new isolates. Some, however, are notably closed, and the example shown here is chlamydia, which is intracellular parasite. So you can sort of make sense that such genomes do not have access to large pool of additional genes. The genes have to come from somewhere. We have seen the data on these open pangenomes, and the genomes come from somewhere. On the other hand, the genome of an organism cannot grow indefinitely. From these very basic observations, we already can surmise that important components of microbial genome evolution are gene gain and gene loss by various forms of horizontal gene transfer. we have done rather extensive quantitative analysis of this process by reconstructing the gain and loss of microbial genes along phylogenetic trees. And we see here what I consider to be a remarkable picture, in the sense that the evolution of prokaryotic genomes is to a much greater extent shaped by gene gain and loss than it is by nucleotide substitution. In these distributions of gene gain and loss rates, as well as the expansion and reduction of gene families by duplication and loss, which was done for a variety of groups of relatively closely related bacteria and archaea, we see these values, which, and here on the logarithmic scale, is the estimated number of gains and losses per nucleotide substitution per site. So we see that by the time a genome accumulates on average one nucleotide substitution per site, there are many and many events of gene gain and loss that will happen. And also quite notably, the ratio of gene loss to gene family expansion, to gene family reduction, and gene gain to gene family expansion rates are large. Gene loss and gene gain are by order of magnitude, if not more common than duplications and loss of gene family members. It's often said, based on analysis of eukaryotic genomes, that evolution proceeds by duplication. This, however, is not really the case in the prokaryotic part of the genomic universe, where evolution occurs more than anything else by horizontal transfer and loss of genes. We have reconstructed the evolution of the pangenomes across the diversity of bacteria and archaea available, that was available a few years ago, and came up with this rather notable picture. So here, these red circles indicate the actual genomic mean genomic sizes across the tree. The green circles indicate the size of the pangenome that can be determined directly from genomic comparison. Those genes that are already available for a given species. You see, of course, that these green circles are, the area of these green circles is larger than that of the red circles, but not that dramatic, perhaps by an order of magnitude at most. And here, these blue circles indicate the estimated size of what we call here a supergenome, which is the limit of the estimated limit of the growth of the pangenome, as we could, for which we could obtain a maximum likelihood estimate from repeated gene gains. And when we have this O here in a large blue circle, that indicates open pangenome, which means that we were unable to obtain a reliable estimate of the limit. The pangenome continues growing at this time without significant signs of saturation. Other estimated sizes are large, but finite, indicating we already see some signs of saturation. this depends on the lifestyle of bacteria, in particular. The free living ones shown in black here oftentimes have open pangenomes, a very large one. The facultative host associated and particularly parasites have often closed pangenome. So here we see, I think, interesting illustration of both dynamics of the evolution of the genome universe and its diversity. So I'll next show and discuss sort of a dissection of the structure of the genomic universe in terms of the gene conservation that reflects the evolutionary regime of different types of genes. So if we use the COG approach and produce such gene commonality plots, which we often analyze, where here on the x-axis is the number of organisms, number of genomes, and here on the y-axis is the number of genes or clusters of orthologs that are represented in exactly this given number of genomes. So here we have a nearly universal core, which is tiny, which is very small, includes essential genes such as those involved in translation processes and others. Here we have a moderately conserved large set of genes, about 2 orders of magnitude larger, which we call the shell, so to speak, around the core, which are conserved in significant number of genomes. And here we have another inflection in this curve. Roughly, it can be approximated by 3 exponential functions, which represents a cloud of poorly conserved, the large cloud of poorly conserved genes and orphans. Very notably, the genomic universe has, speaking somewhat loosely, not quite mathematically, a fractal self-similar structure, in the sense that you see this shape of the gene commonality curve at all phylogenetic lengths. Here we have a comparison of all archaea and bacteria, here archaea only, and here a very shallow comparison of some enterobacteria, Escherichia and Salmonella. You see the same universal shape of this plot, however, even if with different contributions from different components. So we'll discuss in a moment where this universal structure of the genomic universe may come from. Before I do so, however, I just want to bring the attention to the quote-unquote paradox of genomic composition, because in reality there is no paradox, but it's interesting. In each individual genome, as I already mentioned, most genes are highly conserved, as you can see by this red area that represents the core. It's small, but the shell of moderately conserved genes, green here, is large, whereas the fraction of these poorly conserved genes is much smaller. So the genomes are largely dominated by shell genes. But of course, in the pangenomes, the huge overwhelming majority of the genes are the poorly conserved ones and the orphans, which is of quite clear how it happens, because these you happen only once, then they are conserved. These you happen as many times as they occur. So in the sum total, these poorly conserved genes dominate, and that's how the genome universe is organized. So where does the universal fractal architecture of the gene universe come from? A few years ago, with my colleagues, we asked that question, and in order, in an attempt to answer it, compared several models of genome evolution along a phylogenetic tree, which models are listed here in the order of increasing complexity. First is the very primitive neutral model in which all genes have the same replacement rate, the model just one parameter, which implies neutrality. There is no difference which gene you gain, which gene you lose. They are all equal and all neutral. And then we had increasingly complex, perhaps increasingly realistic genomes models that imply selection through either distributed replacement rates or by deriving the replacement rates from a stochastic population model. So we obtain these distributions for many groups of prokaryotes, which show these characteristic gene commonality plots, these sort of U-shaped plots, run the models and see how the predictions of different models fit the empirically observed distributions. You can immediately see that model A, the neutral model, is totally no good. It just does not fit the observed distribution by gross with these red symbols here, by grossly underestimating the fraction of conserved genes. The different models with selection are harder to compare. You cannot quite see the difference visually, but by applying quantitative information criteria, we can see that the more complex and more realistic models that stochastically drives genes from a gene pool is substantially more likely than simpler models with selection. So basically what we conclude is that gene frequency distribution reject a neutral model of genome evolution. So unlike some other universal distributions of genomic variables, for instance, the distribution of paralogous gene family membership, the gene frequency distribution in microbial genomes shows substantial effects of selection. I'll proceed now to a somewhat more sophisticated, more recent modeling of genome evolution that I think, I hope, will provide us with a rough but interesting estimates of the entire size of the entire genome universe. So this was based on the carefully curated database of orthologous genes in archaea, prokaryotic ancestors in which we have specific interest. So COGS for all these archaeal genomes available a few years ago were derived and carefully manually curated and became the basis for modeling genome evolution. When I say modeling genome evolution, I mean tracing the decay of gene content and gene order similarity with different distributions of gene replacement and gene shuffling rate. It's very simple conceptually. We introduce elementary processes such as gene turnover, replacement of genes in genomes, and we also study the order of genes in genomes and introduce gene shuffling, the inversion of segments in genome evolution, and then we construct a genome to start simulated genome in the computer to start with, and allow it to evolve along the empirically obtained phylogenetic tree through these processes. And from this model, we derive the metrics of similarity between genomes in terms of gene composition or gene order. They're similar, as we shall see, and compare actually these different matrices of genome similarity obtained under different models, and compare that to the empirically observed distances and obtain a maximum likelihood estimate that gives you the quality of different models, how well they correspond to the actual observations.
Dr. Koonin, sorry, 2 minutes left.
Yeah. I'm coming to the end of it. So we can see these plots of decay of gene content similarity, the phylogenetic distance. And the interesting point here is that in this type of analysis, we see basically two qualitatively distinct classes of genes. And these are genes with instantaneous turnover, so to speak. We cannot, it drops precipitously immediately, practically at zero evolutionary distance, and that's about 12% of the genomes. And this larger class of conserved genes with distributed rate of loss, but basically the shape of this distribution doesn't really mean. The model with two classes, instantaneous and slow turnover, models best the evolutionary process. And we see just the same, qualitatively at least, it's almost quantitatively same outcome for gene order evolution. So what we see here is a distinct class of extremely fast replaced genes, quote unquote, instantaneously replaced. A little over 10% of the genomes, these basically orphans. By obtaining, so these genes are effectively not shared by genomes, except for the very closest ones. And from this, we can obtain a very simple back of the envelope, but I think important estimate of the size of the gene universe. If there are, on average, about 2,000 genes in a prokaryotic genome, then roughly 200 orphans in each of these genomes, and letters, and very many, from millions to hundreds of billions microbial species on Earth. So we do it very simply. We just multiply this number by the number of species and obtain this total number of genes in the biosphere, in the genomic universe, that may differ from hundreds of millions to trillions, because we do not have a good estimate of the number of microbial species. They are all over the place. But what is very clear that the genome universe is truly vast. So what I have tried to tell you today, that we can now have some evolving, of course, but not entirely meaningless picture of the genomic universe. That genomic universe is highly dynamic. Genomes and pangenomes are exist in incessant gene flux. There is a distinct class of genes that are extremely fast, instantaneously turned over. And then there are core genes with distributed evolutionary race. The sort of neutral evolution of the orphans provides for a huge reservoir of genetic novelty, vast gene universe. Evolution of the core genes is on average adaptive. We can estimate the selection coefficients in other analysis, but selection is on average weak. So I think this is what I wanted to tell you about our glimpses of genome universe. All this was done with a few of my colleagues, members of my lab at the NCBI, above all, the senior staff scientists, Yuri Wolf and Kira Makarov and some former postdocs. Thank you very much for your attention. I will be more than happy to answer questions.